Good morning, everyone. I'll give just a couple more minutes for people to log in, and I just want to say thank you to everyone for attending our first webinar. My name is Jessica Palmer, and I'm the Associate Director at Stemina Biomarker Discovery. And today I'm going to talk to everyone about our quickPredict assay that was recently published, which is a targeted metabolomics-based assay that uses human pluripotent stem cell-derived cardiomyocytes to identify structural and functional cardiotoxicity potential. I'll start with just a little background on Stemina Biomarker Discovery. The company was established in 2007 in Madison, Wisconsin, and we have state-of-the-art mass spectrometry and stem cell culture laboratories. Our expertise is in human stem cell models of toxicity and disease. And over the last 13 years, we've developed a proprietary metabolomics platform that we use for developing toxicology assays as well as diagnostics through our sister company, NeuroPoint DX. Right now, NeuroPoint DX is focused on developing a diagnostic for autism spectrum disorders. And on a general scale, we are focused on identifying biomarkers of toxicity and disease for diagnosis and more precise treatment, drug development, clinical study management, and companion diagnostics. Today, obviously, I'll be focused on our in vitro toxicology platforms. The approach that Stemina uses is a combination of those two platforms, the metabolomics and mass spec and human pluripotent stem cell biology. And when we started developing our assays, we really thought that human pluripotent stem cells offered a unique opportunity to evaluate chemical toxicity using a human endpoint. One of the benefits of using pluripotent stem cells is that they can be differentiated into cells from all types of cells from the body that function similar to their in vivo counterparts, and they really have an unlimited supply because they're able to self-renew in culture. And in particular, they are applicable to high throughput systems, which allow development of rapid in vitro toxicology assays. We've combined this pluripotent stem cell biology with LC-MS-based metabolomics. And we chose metabolomics because the metabolome is really a measure of function, and changes in metabolites can reflect any of the prior they can reflect changes in gene expression, changes in transcriptomics, as well as changes in proteomics. And so they're really, considered to be closest to the phenotype and more sensitive than genomics, transcriptomics, and proteomics. And through using these two endpoints, the pluripotent stem cells and the LC-MS-based metabolomics, our goal is to develop in vitro toxicology assays that would allow our clients to understand compound risk profiles faster, looking at multiple exposure levels, using a minimal amount of compound, and all at a cost savings compared to the standard in vivo toxicology tests that are used during the preclinical phase. To date, we have developed two exposure-based assays for exposure-based prediction of toxicity potential using this targeted metabolomics and pluripotent stem cells or cells that are differentiated from them. And so those are our DevTox quickPredict assay, which predicts the developmental toxicity potential of a new drug or a chemical, and will be the focus of another webinar by Stemina being held next week. And also, as well as the Cardio quickPredict assay, which is what we'll be focused on today. All of the research that I will discuss today was recently featured in the April 2020 issue of Toxicological Sciences, which was a joint publication between Stemina and our collaborators at UCB, Jean-Pierre, Valentin, and Vitalina, Gryshkova. And we were really excited about this new publication as it was featured under the Emerging Technologies, Methods, and Models section for Toxicological Sciences, and really is the culmination of many years of work. So predicting, as many of you may know, predicting cardiotoxicity can be a real challenge during drug development because cardiovascular toxicity, and because of that, cardiovascular toxicity is still a major cause that leads to compound attrition throughout the drug discovery and development process, as well as leading to withdrawal of FDA-approved drugs from the market. And part of this is because there is such a broad range of drug-induced cardiotoxicity as it can affect all parts of and functions of the cardiovascular system. These effects can be separated into functional or electrophysiological or structural. And by functional, really, I mean that it's something that's where a drug causes a main effect on the electrical or mechanical function of the heart, whereas with structural, the main effect is on general cellular processes or morphological damage to the cardiomyocytes, such as changes in cell viability, metabolism, mitochondrial function, oxidative stress, and results in things like cardiomyopathy. The current preclinical cardiovascular toxicity testing methods are really heavily focused on this functional assessment. And there are really limitations to the standard in vivo models used due to numerous species differences in the ventricular repolarization and drug response. And because of this, there are a number of ongoing initiatives that are aimed at addressing these limitations for proarrhythmia, but there's really been an unmet need to test for structural cardiovascular toxicity early in drug development. And there's really a limited number of in vitro assays to determine the potential for structural cardiotoxicity. And I forgot to mention at the start, but if anyone has questions during my webinar, please type them into the question and answer box, and I will answer them at the end. So because of all of these limitations and this unmet need for structural cardiotoxicity testing, we decided to develop our quickPredict assay. And the goal was to develop an assay that was not just focused on functional cardiotoxicity, but also able to identify structural cardiotoxicity. And so to do this development, we really had two main phases. The first was the biomarker discovery phase, where we were looking for what biomarkers were changing in response to a drug. And then the second was to develop a targeted platform for rapid prediction. The assay was developed and uses the iCell cardiomyocytes squared from Fujifilm Cellular Dynamics. And throughout this entire process, we used a total of 81 well-characterized drugs that had well-known cardiotoxicity outcomes. 52 of them were cardiotoxic drugs and 29 of them were non-cardiotoxic. We then took those cardiotoxic drugs and separated them by their main effect type, whether that was a structural effect, functional effect, or drugs that caused both structural and functional effects, which are referred to as a general cardiotoxin. This set of compounds included a wide range of cardiovascular and non-cardiovascular drugs, including channel blockers, anti-neoplastic, antiviral, COX-2 inhibitors, receptor antagonists and agonists, and tyrosine kinase inhibitors. And so the goal of the first phase of the assay development was to identify predicted metabolites that were secreted from or consumed from the cardiomyocytes that could discriminate cardiotoxicants from non-cardiotoxicants independent of changes in cell viability. And then once we had started to identify these biomarkers, we wanted to confirm that they were reproducible in multiple replicates over time, including multiple lots of the cardiomyocytes from cellular dynamics. And so to do this, we used 2 metabolic profiling experiments. The first was a concentration response profiling experiment. And during this time, we looked at an 8-point concentration curve for 50 drugs and used that as an initial development or identification of predicted biomarkers. But the main point was to really identify a non-cytotoxic concentration that altered cardiomyocyte metabolism. And we then selected and used that non-cardiotoxic concentration. And for the second set of experiments, which were a single exposure metabolic profiling experiment. During this phase, we used 66 drugs, and they were evaluated in 2 independent replicates at a single exposure. And we were able to, and what we did is we measured changes in the secretome using untargeted metabolomics methods multiple times. And so when I refer to the secretome, it's not we're not taking the cells and measuring the metabolites inside of them, but instead we're measuring how they're interacting with their media. So what are they secreting into the media and what are they consuming with the media? And we are most interested in is which of those metabolites are changing in response to a cardiotoxic drug, but not changing in response to non-cardiotoxic drugs. So through this, we identified a total of 156 secretome features, because at this point, they're really just a mass and a retention time. They're not, they haven't been identified as known metabolites. That's part of the untargeted platform we use. And then we went through and selected the most predictive features from these 156. So we wanted to identify which features were able to discriminate cardiotoxins from non-cardiotoxins with a high positive predictive value. And so once we were able to narrow it down to those most predictive features, we looked at combinations of those features to further narrow it down as to which ones were going to be complementary to each other and would be amenable to confirmation and developing this targeted platform in phase 2. And we used a 4-fold cross-validation statistical method to do this. So we took from those 13 features, we were able to narrow that down to really 6 or 7 features that we took forward into phase 2, where the goal of this phase was to develop an exposure-based targeted metabolomics assay for a rapid prediction of cardiotoxicity potential. And so we were able to confirm the identity of 6 of the most predictive features, which were arachidonic acid, thymidine, 2-deoxycytidine, lactic acid, alanine, and N-acetyl-aspartic acid. And so we took these 6 features and developed a optimized LC-MS method for them that really allowed a 4-fold increase in throughput. And this is, when you do an untargeted metabolomics analyses, you have long LC-MS runs, and it can take multiple days to do a single 96-well plate. And so we wanted to really get that down to a run where we could be analyzing multiple plates in a single day. And so again, once we developed this optimized platform, we ran all 81 drugs with the new LC-MS method in an 8-point dose response curve, and then used those results to set predictive thresholds for each metabolite, and then again used fourfold cross-validation to assess the performance of the metabolite combinations and select final models for that final, the final assay model that was then taken to market as our quickPredict assay. And the final prediction model included 4 metabolites: lactic acid, arachidonic acid, 2-deoxycytidine, and thymidine. We found through developing the LCMS phase, alanine was not able to be measured reproducibly or meet all of our standard criteria in the same method as the other 5 metabolites. And so, and alanine also had overlapping predictivity with lactic acid. So that one was dropped from the model. And then when we did the fourfold cross-validation, we found that the model with these 4 metabolites performed higher than the model with all 5 metabolites, indicating that N-acetyl-aspartic acid really was not adding anything to the prediction beyond what we were already getting. So what does the final process look like? So the final process is we take the cardiomyocytes squared from cellular dynamics, and we'll plate them into 96-well plates. Every plate contains controls to look at whether or not the cells are metabolizing these metabolites in a manner that's consistent with how the assay was developed, in a manner that's considered normal. And then we'll expose the cells to drug for 72 hours with test article changes every day. After that 72 hours, we'll collect the spent media for analysis using the targeted platform we described. And then we'll do a cell viability analysis on the cells. But the beauty of using the spent media is that it also allows for other downstream analysis of the cells, and it doesn't destroy the cells upon collection. So we could collect those cells for gene expression experiments, or we could also do other electrophysiological assays while we're conducting these experiments for our quickPredict assays, such as MEA or impedance-based assays. So and then we'll take the data from the cell viability and the LCMS analysis and analyze it, looking at the dose response profile for each of the metabolites, and really combine that into our final quickPredict prediction model, which I'll go into more detail. So the prediction model is the composite of those 4 metabolites and cell viability. We use viability and viability over lactic acid as the predictive ratio for lactic acid, and viability over arachidonic acid instead of using the lactic acid and arachidonic acid by themselves. And this is because both of these are media components that are not changing when they're not treated, but when the cells die, you'll see, because they're not changing, if you have a compound that's responding to them, where the cells are responding, you'll see this increase, and then if the cells die, they decrease back to 1, which makes it difficult to interpret the data. So we normalize those to lactic acid, or normalize those to viability. Each of these metabolites have their own upper and lower prediction thresholds that was set with based on the response at 10 times the Cmax for all 81 drugs. And each of these thresholds could be optimized for balanced accuracy or sensitivity, and I'll go into that a little later in the presentation as to why that may be important. So after you have these prediction thresholds, how do you go from 4 independent metabolites to a composite model for a single prediction of cardiotoxicity? So what we do is we look at the change in response at a given concentration for each metabolite and normalize that to the metabolite-specific cardiotoxic response threshold. So if the response of the cells is closer to the upper threshold, the upper prediction distance is determined by dividing this response value here, which would be about 3.25, by the metabolite threshold. Whereas with the lower prediction distance, you have the metabolite threshold, which depicted here is about 0.5, and you would divide 0.5 by the metabolite response, which is about 1 in this case. And so the composite prediction distance for that composite model is really based on the maximum response or the maximum prediction distance at each concentration. And so we take the values from each of the metabolites at each concentration, and the value that is the largest is the value that goes into this composite prediction model. And this allows it to really, the composite prediction is then reflective of the most sensitive ratio at any given concentration. Excuse me. And then the other thing I should note is the composite model as well has its own threshold of predictivity. And so a value of 1 would mean that the metabolite response was right at that prediction threshold. And so you could, again, optimize this threshold for sensitivity or balanced accuracy, depending on how you would want to use it. So then we can take, for the final prediction of cardiotoxicity and the prediction of the concentration where a compound is going to be cardiotoxic, you can interpolate where this dose response curve for the composite prediction distance crosses what we've termed the cardiotoxicity threshold, which is the optimized threshold for the composite prediction model. And you can figure out at what concentration is this dose response curve crossing that threshold and use it to determine where a drug is more likely to be cardiotoxic versus non-cardiotoxic. So, concentrations that are lower than this CTP are the non-cardiotoxic exposure range, whereas concentrations above that would be your cardiotoxic exposure range. So to evaluate the performance of the model at this early stage during development, we looked at two different two different optimized thresholds. Because as I mentioned, these metabolite-specific prediction thresholds can be optimized to meet the needs of the end user. So we looked at sensitivity optimized thresholds, which would be beneficial for application during early drug discovery, when it's more important to accurately predict true positives, and you're not as concerned about false positives. Whereas you could also optimize it for balanced accuracy, where you're going to have higher specificity in an effort to, if you wanted to apply this a little bit later on in the drug development pipeline, when you want more highly specific models. And so we used the response at 10 times the Cmax to score the classification and the performance of the prediction models. And so if a true positive would be if the response falls into, the response at 10 times the Cmax falls either below the lower threshold or above the upper threshold. A true negative would be when your response is, around 1 or falling in between these thresholds. A false negative would be a positive compound that did not show a response at 10 times the Cmax, where it fell into these cardiotoxic zones. And a false positive would be something that should be negative, but it is showing a response at 10 times the Cmax. And so when we looked at the final model and the retrained thresholds with the balanced accuracy optimized model, we had an overall balanced accuracy of 86% in the composite model. And you can see that really these metabolites that go into that composite model are really complementary, because your sensitivity for any given metabolite by itself is less than 70%. But when you combine those four metabolites, you get a sensitivity of 83% in the composite model, and still are able to maintain excellent specificity of 90%, giving you a very high positive predictive value. So you're confident that a drug that is predicted to be cardiotoxic with this assay is likely to be cardiotoxic in the real world, or in vivo, I should say. And when you look at the different classes of cardiotoxicity, you can see the functional cardiotoxins were identified with 93% accuracy, and lactic acid was really the most predictive for functional cardiotoxins. And structural cardiotoxins were identified with 63% accuracy, and compounds that had both functional and structural cardiotoxicity, things like sunitinib and mitoxantrone, cisapride, things like along those lines, really were identified as well, and they kind of were more spread out across the four metabolites. Again, when you train this model and train those thresholds so that they're optimized for sensitivity, you will still maintain an excellent balanced accuracy of 85%, but your sensitivity goes up to 90%. But one of the downfalls of that is you are going to start seeing a decrease in specificity. 79% is still very good, but obviously not as good as 90. And but with this, you're able to identify 100% of the functional cardiotoxins, 75% of the structural, and 95% of the general. Now, we recognize that during early drug development, you may not know what the Cmax is going to be. And so if you're going to apply the assay and don't want to use a Cmax, you can use that quickPredict, the cardiotoxicity potential concentration as a signal of the cardiotoxicity of the drug, or you could set a threshold, a concentration threshold for predicting, excuse me, you can set a concentration threshold for predicting positive or negative. So this is just an example that I discussed that was in the discussion of our paper, where if you apply a threshold of, say, 30 micromolar, you can maintain that balanced accuracy and sensitivity with a slightly decreased specificity. But again, you're able to identify your functional, structural, and general cardiotoxins with very good accuracy. So why are these four metabolites so predictive? Why are they important for cardiotoxicity? And really, all of them are involved in biologically relevant pathways that have been shown to be important for cardiotoxicity. And one thing that's really important to point out is these are all changing, and all of these predictions are made independent of any changes in cell viability. The fact that they are affected by such a wide range of compound classes and compound mechanisms, it implies that there really are some common metabolic pathways of toxicity that are shared by many cardiotoxicants. And these four metabolites are known for having key roles in modulating oxidative stress and mitochondrial function and replication, which, as I mentioned, are known mechanisms of cardiotoxicity. And I'm going to show you the different response types that we see, and there is a very broad range of responses observed for each of these metabolites, indicating that each biomarker can really is picking up cardiotoxicity through multiple mechanisms. So lactic acid is present in the media, but it's not utilized by the cells under normal conditions. And so when you see an increase in lactic acid, that means the cells are secreting lactic acid into the media. If you were to see a decrease in lactic acid, that means the cells are actually consuming lactic acid from the media, which the reference controls are not doing. So the most common effect that we do see is an increase in the media, which, as I mentioned, was really common for the functional cardiotoxicants that we tested. As I had mentioned, when you have changes in cell viability, you're going to see a biphasic response, where when the cells start to die, this increase drops back down to 1. There are a few compounds that actually will decrease lactic acid relative to the controls, such as amphotericin B. But this wasn't as common as this increase that we're observing. And really what this does is it provides insight into the mitochondrial energy metabolism of the cardiomyocytes. And its increased secretion reflects a shift from oxidative phosphorylation, which is how adult cardiomyocytes get their energy, to anaerobic respiration and glycolysis, which is really a hallmark of mitochondrial dysfunction. And elevated levels of lactic acid in the blood have been associated with cardiotoxicity, especially during cardiac ischemia and heart failure in vivo. Arachidonic acid is also a media component that's not used by the cells. So again, when you see an increase, it indicates that the cells are secreting it. This one, unlike lactic acid, where you had kind of one common response across the drugs, you can see a multitude of responses. So there are a number of drugs that cause the cardiomyocytes to secrete arachidonic acid, and then there are drugs that will cause the cells to increase their uptake of arachidonic acid, which is noted by a decrease here. And again, when you have changes in cell viability, you're going to see a biphasic response. And then there are some drugs that cause this increase at lower concentrations and a decrease as you get higher in concentrations, indicating, again, there are multiple mechanisms at play here. Arachidonic acid is involved in a number of different biochemical pathways. It's important for lipid transport, lipid metabolism, and fatty acid metabolism. And activation of the arachidonic acid cascade due to a redox state unbalance has been shown to contribute to the pathogenesis of cardiovascular disease in vivo. A number of the cardiotoxic compounds that we tested are known to modulate CYP2J-mediated arachidonic acid metabolism, leading to increased levels in the plasma in vivo. And so it's, they're changing how arachidonic acid is broken down and whether it's broken down into cardioprotective or cardiotoxic metabolites. And one example of that is doxorubicin has been shown to have that effect in vivo. And the thiazolidinedione, such as rosiglitazone, has been shown to increase arachidonic acid release from the cell membrane, which would be reflective of it as an increase in arachidonic acid metabolism. And here in panel A, as an example, is the response we observed to rosiglitazone in the assay, demonstrating that the response we're observing matches what was observed in vivo. 2-deoxythymidine, I'll talk about kind of together because they have similar mechanisms, or similar functions, I should say, and they are both secreted by the cells into the media. And They will increase and decrease with toxicity. So again, indicative that there are multiple mechanisms leading to differences or changes in the levels of these two metabolites. For thymidine, the most common response you see independent of cell death is an increase in thymidine. So you see an increase in thymidine secretion. And typically when you see a decrease, that's occurring when you have cell death because there are fewer cells to secrete thymidine. In contrast, 2-deoxycytidine, we saw a decrease when the cells were treated with specifically different neoplastic drugs, and saw a decrease in 2-deoxycytidine secretion independent of changes in cell death. There were a couple of drugs such as clozapine where you saw an increase, which then decreased when the cells died. And again, something like pergolide, where you're seeing, again, this multiphasic response where it's causing a decrease in 2-deoxycytidine secretion, followed by an increase at higher concentration. These are two of the principal nucleosides of DNA, and they're components of the pyrimidine metabolism pathway. And changes in these two metabolites really reflect inhibition of mitochondrial enzymes that are necessary for mitochondrial DNA synthesis and replication. And mitochondrial DNA damage in cardiomyocytes has been shown to be a mechanism of doxorubicin-mediated cardiotoxicity. And many of the antiviral and anti-cancer agents known to cause cardiotoxicity are nucleoside analogs, such as zidovudine and fluorouracil. So how can you use all of this data together? And how do you predict functional versus structural? And so if you look at the response of the, so you can use that composite model to really predict whether or not a compound is going to be cardiotoxic or not. If you want to look at the likelihood of functional versus structural effects, you're going to want to look at the individual metabolite response and understand which compounds are changing which metabolites before any changes in other ones. So as I had mentioned, lactic acid is really an indicator of functional cardiotoxicity, and you can see beprudil and cisapride are examples of two non-cytotoxic functional cardiotoxins that that caused an increase in lactic acid at concentrations lower than where you saw changes in the other three metabolites. Whereas arachidonic acid, thymidine, and 2-deoxycytidine were really not changing with most of the functional cardiotoxins, or changing at much higher concentrations with them. And these are more indicative of structural cardiotoxicity. So things like imatinib and rosiglitazone cause changes in arachidonic acid metabolism at lower concentrations. Sunitinib and Herceptin or trastuzumab changed thymidine at lower concentrations than the other metabolites. And two examples that changed 2-deoxycytidine at the lowest concentrations are amsacrin and doxorubicin. And so you can really use this data to understand differences between the compounds, as well as within a compound class, such as the tyrosine kinase inhibitors, and to understand differences in mechanisms and their cardiotoxicity potential. So an example with the kinase inhibitors is, the challenge is really predicting the likelihood and type of cardiotoxicity with these drugs. And they all have, multiple mechanisms and inhibit different kinases. And you can see that these drugs are really inhibiting or changing different metabolites at different concentrations. So as I mentioned, sunitinib here is changing thymidine, whereas dasatinib and erlotinib are changing 2-deoxycytidine, and imatinib and lapatinib are changing arachidonic acid at the lowest concentrations. And crizotinib, which is known to cause functional cardiotoxicity, is really showing a response in the viability over lactic acid response ratio at the lowest concentrations. Whereas axitinib, which is considered to be cardiosafe, isn't changing any of the metabolites. And so you can use all of this data to really prioritize which candidates should undergo further safety pharmacology testing, which ones you would want to look at with different in vivo studies to really understand the mechanisms better, and prioritize the order of doing these assays. And so in summary, the combination of 4 metabolites is able to predict cardiotoxicity with high accuracy at therapeutically relevant concentrations. Each metabolite in the model can identify multiple mechanisms of cardiotoxicity, and the predictive model can be used to determine a concentration where a compound shows the potential for cardiotoxicity. And really, one of the beauties of this method is that it can be combined with other assays or endpoints such as MEA or impedance for a more comprehensive understanding of a compound's cardiotoxicity liability. So with that, I'd like to acknowledge my team at Stemina that has helped me develop this assay and done a lot of the groundwork to get it up and running. Alan Smith, Michael Caldwell, Mike Ludwig, Delray Sugden, Daniel Brass, Fred Kirchner, who's our Director of Toxicology, Robert Burrier, who is our Chief Operating Officer, and Elizabeth Donley, who is our CEO. And with that, I'll take any questions. So the first question is, does your culture contain glucose, and what was the concentration if it was added? The media does not contain glucose. We use the standard cardiomyocyte media from Cellular Dynamics, and I can't divulge all of the components of their media, but I can say that glucose is not the carbon source in that media. And there's been a number of publications that show when things like galactose are used instead of glucose, the cardiomyocytes actually have a more metabolically mature phenotype compared to when glucose is contained in the media. And another question is, for lactate, does it decrease in concentration? As I discussed, it typically will actually increase. And where does the lactate come from? I believe the lactate in the media is actually coming from the serum, because the media does contain serum, and you can see similar levels in the media controls in a plate. So wells that do not have any cells, the lactic acid levels in there are the same as our reference controls. And so the hypothesis is, since it doesn't appear to be added, it's not added by Cellular Dynamics, and it's not in the basal DMEM that's used to make the media, we're pretty sure it's coming from the serum. And next question is, do you see a lot of differences between stem cell lots with the reference drugs? And the answer is no, we did not. We used, I can look up the exact number, but I think we used at least 5 or 6 different lots throughout the development of this process, and different lots during different phases of the assay. And even we did not see any differences in the metabolite response or the concentrations really where we observed an effect. And I would also note that the very first step of the assay development, where we did the initial dose response metabolic profiling experiment, was actually done with the original iCell cardiomyocytes. And then we did a study to look at whether or not the cardiomyocytes and the cardiomyocytes squared had similar metabolic profiles, and show that they were equivalent. because at that point, after we had done that first study, the iCell cardiomyocytes squared became available. And so we were able to show that they were the two cells were equivalent in their metabolic response and did not show any differences in how they responded to compound. So how does lactic acid increase? And that's, it's a challenging question. I think that the hypothesis we have is that it's increasing because the cells are switching from oxidative phosphorylation to glycolysis, leading to an increase in lactic acid production, which is leading to an increase in lactic acid secretion. Are there any other questions? And so another question. Did you compare your assay with some tox assay kits on the market like MTT or LDH gas-based? We did not do MTT or an LDH or gas-based. We did not do any of those other assays during development. What we did was a cell titer floor viability assay from Promega, which measures a live cell protease. And so we didn't do some of the other cytotoxicity kits. What we wanted to look at, what we looked at was the number of live cells left in the dish. And that's an assay that we've used in the lab standard for all of our development of the different in vitro tox assays that we offer. And we've looked at some of the other assays early on during assay development, whether we wanted to use that one versus something like Alamar Blue or the MTT assay, and we found that really the cell titer floor assay was a more accurate predictor of cell viability or cell number, and was the easiest to perform and most reproducible, I should say. And so then the next question would be, do you mean lactate is produced from galactose? And I would say that not necessarily. We haven't done any studies right now that to look at and confirm our hypothesis of how the lactic acid is being produced. And I would want to confirm with our COO, who is has a more knowledge on the lactic acid metabolism pathway than I do. And so I can follow up with him to see if I can get a more in-depth answer as to why we think lactic acid is increasing and how it's being produced, because there isn't a direct link between galactose and lactate. So I'll look into that and can send send a response to you, John. Did you add pyruvate? Pyruvate, I believe, is in the media. It's actually one of the metabolites that was not included in that final prediction model, but it is one that we confirmed as being present and changing in response to, we confirmed it during the development of the assay. It just didn't make the final cut for predictivity. And so pyruvate is there and it is changing, but I'd have to look back and see whether it's something that was being secreted by the cells or something that was added to the media. If there are no other questions, my contact info is here on the screen, and I believe that we'll be sending out a link to this presentation afterwards today, and so you should have that if you come up with additional questions that you'd like to ask. Fatty acids. We did find some fatty acids that were changing, and but they weren't measured as well with the methods we were using, and they weren't predictive across the board. So I think arachidonic acid was the only link to fatty acid metabolism that we found that was predictive of cardiotoxicity. And also I'd like to just let everyone know that on this, I do have a slide here for everyone that does have links directly to our publications. If you're interested in reading on the targeted, reading our new publications that came out in Toxicology in April, and as well as my email. So if you have questions, you can follow up. So, thank you everybody for attending, and I hope you have a great rest of your Friday and Memorial Weekend if you're here in the US.